Hello everyone, this is Jason from STEM Nerds. Thank you so much for being here. We've got a really important video today because we're going to be talking about how microbes grow. And we really can't understand why microbes cause infection, why they help us in an environment, and do all the good and bad things they do without understanding how they grow. And so in today's video, we're going to learn a lot of words. We're going to learn how to classify microbes based on their environmental parameters. We're going to learn how to quantify microbes and the tools that we use to do that. Thank you so much for being here. So first things first, microbes grow and divide via binary fission. This is a lot different than mitosis or meiosis, if you've learned those processes in the past, because microbes, bacteria, are prokaryotic. They lack membrane-bound organelles. They lack a nucleus. They have one circular chromosome, and because of that, they are a simpler form of life. And basically what they do in the very, very short version of binary fission is they copy that circular chromosome. They may copy some plasmids if they have them. And then the cell splits in two. And so ideally what we have here, when this happens, is we have a cell. The offspring is genetically identical to the parent cell. This very well may be one of the most important things that we go over. I know it's not flashy or anything, but I want to show you something that is very, very important. And it really drives home the point of why it's so important to control microbial growth, something that we're going to talk about in a future video. So we have a growth curve here. And this growth curve, don't let the math scare you. It's not that bad. I'm actually going to show you how to do all this in a second. But basically what we have is we can figure out the number of cells that we're going to have at a future time. If we know the number of cells we're starting with, the amount of time they're going to be growing, and how long it takes them to reproduce. And so I've given you 4 examples here. And I want to drive home the point on why it is so important to control microbial growth. And so here's how we figure this out. And I'll give you the first one. So in number 1, we're starting with 3 cells. They have a generation time of 30 minutes, and they're going to divide for 8 hours. And so we look at that formula. So that n to the naught right there, that n looks like NO, that's going to be 3. Right? That's the number of cells we're starting with. Times 2, 2 is a constant. The n is the number of divisions over a given period of time. All right, now this is where a lot of people get this all messed up, so let's pay attention here. Okay, so we've got 3, number of cells we're starting with, times 2, what is n? Well, let's take a look at our question number 1. We have 8 hours, and we know the microbes can divide every 30 minutes for 8 hours. So how many 30-minute time periods is there in 8 hours? Well, there's 16. So the way we would plug that formula in is it would be 3 times 2 to the 16. And we calculate that out, we get the number of cells. And we can do that for all 4 of these. And so I would highly recommend, try it out, see what you get. So I know the suspense is building and we need to figure out, okay, well, what does this all mean? So let me show you here again. So I did the first one, 3 times 2 to the 16th power. And what we have here is after 8 hours, we went from 3 cells. Now we have 196,608 cells. And so you can see that is a lot of growth. And so the first thing we have to know is that when microbes are growing and dividing, it is exponential growth. Every time they grow and divide, they double the number of cells in that population. So it's a very powerful thing. That is why we want to control microbial growth in and around our food and our water, what's growing on countertops and that kind of thing. But what's even more powerful is, let's just assume that question number 1 is kind of like a baseline. But now I want you to look at what happens. Question number 2. We had 3 cells, so same exact number of cells we're starting with. They're going to divide the same amount of time, so 30 minutes. Right? Their generation time is the same, I should say. However, we've changed the amount of time that they're allowed to grow. In fact, we've tripled it. We've gone from 8 hours to 24 hours. But the thing you need to see is, again, we did not just triple the amount of cells relative to question number 1. No, it's not even close. Here we have 8.4 times 10 to the 14. And so it's 8.4, and you've got 13 more zeros after it. That is a whole lot of cells. So what's changed? We allowed the microbes to grow for a longer period of time. That 24 hours had a massive impact, and that is point number 1 of this little exercise that we're doing. We need to control how long microbes are allowed to grow and divide, because the the growth is not linear. It is exponential growth, which means every time that division happened, we double the number of cells, and we have way more cells here. So again, if we're dealing with food processing, if we're dealing with water, if we are dealing with biocontamination, if we're dealing with the human body, we have a critical time to control these microbes. It's absolutely critical. So let's go back to question number 3 here. Not so extreme. So in this question, the only thing that's different compared to question number 1 is we just double the number of cells we're starting with. And so instead of having 3 cells, we have 6, but we still let them grow for 8 hours. Generation time was 30 minutes. And so we do the math, and interestingly enough, we started with twice the number of cells. We doubled the concentration of the cells at the end of 8 hours. And so again, that's point number 2. Point number 2 is you always want to try to control the number of microbes in an environment from the beginning, because the more you have to start with, the more you're going to have at the end. And that is kind of a linear relationship, right? So we doubled the number of starting cells, we doubled the ending concentration, but it's still important to control the number of cells you're starting with. Okay, question number 4. This is also really important. It's going to bring home point number 3. We started with 3 cells, so we're back to our original number of cells we're starting with. The difference here is that the generation time is 20 minutes and not 30 minutes. And so our cells are a little bit happier. They're in optimal conditions, but we've only got the amount of time they're growing for 8 hours. Really what we're doing here, and what I want you to see, is compare the first scenario with this last one. And again, what we're seeing here is generation time matters a lot. In fact, it matters a lot more than even the beginning number of cells you're starting with, because here, 5.03 times 10 to the 7, we now have millions of cells. And the only thing that changed is we went from 30 minutes down to 20. And so we made the cells a little bit more comfortable, and they grew a lot more. And again, we see a massive difference, exponential growth. And so we've learned something. One, we've learned try not to leave microbes growing for long periods of time. The sooner that you can get rid of microbes, the less concentrated they're going to be. 2, control the number of cells you're starting with, because realistically, you're not usually just starting with 3 cells or 6 cells or 10. You might be starting with hundreds or millions. Third thing we learned is that generation time definitely matters. When we went from 30 to 20 minutes, that is huge as well. And so generation time and certainly the amount of time that they're growing are going to have the 2 largest impacts. So what makes microbes comfortable? Well, there are environmental factors that we look at when we are looking at how microbes grow. And there are words that we use to describe microbes in certain environments. This first one here is how we describe microbes based on how they grow at certain temperatures. And so we can see here we've got this word psychro. Psychrophile, psychotroph. And basically what that means is these are microbes that grow in cold environments. And what's useful for us to understand is these are microbes that might spoil food in a refrigerator. Now, most microbes are not going to survive under the freezing point, right? So as soon as something freezes, a lot of times microbes will actually lyse. But you're going to see here for the psychrophiles that we have them growing at less than 0 degrees Celsius. That's weird because water freezes at 0 degrees, and I just said you need liquid water. They can't grow in ice. However, if you start looking at microbes that are growing near the North and the South Pole, you have a lot of salt in that water, and that water can actually go below 0 degrees Celsius and still remain liquid water, and so that's why we're seeing those there. And so psychro, the main point is they're growing in colder environments. They are usually responsible for food spoilage in a refrigerator. They can certainly, especially the psychotrophs, be very happy at room temperature. They don't do very well in the body, right, in the human body. The mesophiles, on the other hand, they do. Mesophiles here, these numbers here are a little bit skewed. Mesophiles typically have an optimal range of around 37 degrees Celsius, which is the human body temperature. And so the thing we should know about mesophiles is those are typically the microbes that we worry about when it comes to causing human disease. A lot of them are human pathogens because their optimal growth temperature is the same as our body temperature. And then we get into the thermophile, hyperthermophile, These are heat-loving. Phyl, by the way, anytime you see that word phyl, it means to love. So these are heat-loving microbes, the thermophiles, the hyperthermophiles. They're going to be in extreme environments. Mostly they're the archaebacteria. You're going to see them, especially the hyperthermophiles, around thermal vents at the bottom of the ocean where there's volcanic activity, where that water can become superheated and, yes, be above the boiling point, which is 100 degrees Celsius, and go above that because of the high pressure, the salt concentrations, and everything that's going on down there. And so the thermophiles, heat-loving microbes. This next environmental factor is really important and something we're going to come back to in a future video. And it's how we classify microbes based on whether or not they require oxygen. And what we have here are what are known as some nutrient agar deeps, like it's deep. And basically what this is it's a food for bacteria that's a semi-solid. It's almost like a gel, and only it's agar. And so what happens is there is an oxygen gradient within the agar itself. The closer you are to the surface, the more oxygen is available. The further down in that tube towards the bottom, the less oxygen, or i.e. almost no oxygen, is available. And so on the left-hand side, we have these microbes called obligate aerobes. They require oxygen. We, as human beings, are obligate aerobes. Well, there are microbes just like us. They need oxygen or they're going to die. Now, in a future video, we're going to talk about the importance of oxygen and why it's necessary. But for right now, we just need to know obligate aerobes, they need oxygen or they cannot survive. And so if we were to grow these microbes in a nutrient agar tube, like you're seeing here, they usually just grow at the surface where the oxygen is available. Now, you're going to see the next one over from the left is the microaerophile. Micro means small. Aerophile, a little bit. Aero, oxygen, phile means to love. So they love a little bit of oxygen. And you're going to see here, so if the typical oxygen concentrations are around 20 to 21% in our atmosphere, the microaerophiles might be somewhere in the upper single digits or so in terms of percentages. So they need some oxygen, but not a lot. Now, one of the reasons why this is so important is because certain microbes have enzymes that detoxify oxygen radicals, so an oxygen that has an extra electron. And believe it or not, when an organism is in the presence of oxygen, oxygen can actually become dangerous. It can become dangerous for us too, but we have ways to detoxify when oxygen becomes dangerous inside of a cell, as do microbes, certainly that are obligate aerobes. But these microaerophiles, they lack these enzymes. One of the most important for bacteria is an enzyme called catalase. It's a lab that we do in microbiology to determine whether or not a microbe has catalase. It's not the only enzyme, and so I'm going to say catalase-like enzymes when I'm describing this thing that we're seeing here. But a microaerophile has these enzymes that are like catalase, but not in high concentrations, and so they can get rid of some of the toxic byproducts of oxygen, but not all of them. And so when the concentrations get too high, they die. But they need some, so they can't get too low. So they're very picky. I call them the very picky oxygen consumers. Okay. The middle tube, obligate anaerobe. These microbes, this is going to sound so strange to someone who's never studied microbiology before, but there are actually living things that are killed in the presence of oxygen for the reasons I just explained. The obligate anaerobe cannot be in the presence of oxygen or it will die. And the reason for that is it lacks the catalase-like enzymes that detoxify oxygen. And so it's in the presence of oxygen, those oxygen radicals build up, destroys the cells. And so you're going to see them in areas where there is no oxygen. Again, now why is this important? Because the obligate anaerobes fill a very specific niche within our environment, something that we talk about in a future video, but we usually find them like really deep in sediment and aquatic environments. We find them deep in mud, so places where there is no oxygen. These last two at first look very, very similar. We have what's known as a facultative anaerobe, and then on the right-hand side, we have an aerotolerant. It should say aerotolerant anaerobe there. Both of these microbes, the facultative anaerobes and the aerotolerant anaerobes, can survive in the presence of oxygen. The facultative anaerobe is perhaps the most versatile microbe of any of the microbes that we talk about here today when it comes to oxygen concentration requirements. And the reason for that, the facultative anaerobes prefer oxygen, and that's why you see them growing at the top. But look what happens. As you go further down and oxygen is unavailable, they're still there. but not at the same concentration. That's why you don't see as many dots towards the bottom. And the reason for that is the facultative anaerobe uses oxygen if it's available, but if it's not available, it can switch to a process called fermentation. And again, in a future video, we talk about all of this stuff. For right now, we just need to understand facultative anaerobes prefer oxygen. If it becomes unavailable, they can switch to fermentation. And yes, they do possess the enzymes that detoxify oxygen radicals. How is that different from the aerotolerant anaerobe? So you can see it's growing everywhere. The aerotolerant anaerobe is not killed by oxygen. However, it doesn't use it. So basically, it's indifferent to oxygen. The aerotolerant anaerobes are usually obligate fermenters. In other words, the only process that they can do is ferment. They usually lack the structures that are involved in using oxygen, what's known as an electron transport chain. Again, a process we'll get to later in a future video. But what you need to know right now is the aerotolerant anaerobes are indifferent to oxygen. It can be there, it can be missing. It doesn't matter. They don't use it. They're not killed by it. They are strict fermenters. And so these are ways that we classify microbes based on oxygen requirement. The reason that's so important, once again, is the obligate aerobes, those microbes can be where we are. As long as there's oxygen available, they can grow. Microaerophiles are a little bit picky. Obligate anaerobes are usually found in areas where we cannot be found in as human beings. Facultative anaerobes and the aerotolerant anaerobes, these guys are often associated with us, especially in the human large intestine and other parts of the body. So I mentioned these obligate anaerobes, and they're kind of tricky. In a lab, if you want to grow them, you have to use specialized equipment. This is called an anaerobe jar, and basically what it does is you put a packet in there that reacts, takes all the oxygen out of that, you seal it tight so there's no oxygen, and that's the way you grow them. And so we used to do that in one of the labs that I was teaching at. We would isolate anaerobic microbes from the soil, and the way you had to do that is you took your soil sample and you put it on those plates, those nutrient agar plates, and then you sealed this thing up. It removed all the oxygen, and then they would grow. But you expose them to oxygen, they would all die. And so we've talked about how to classify microbes based on oxygen requirements. We've talked about temperature. There's a couple more, not nearly as complicated. One of them is pH classification. We can classify microbes based on where they're growing in terms of their pH. An acidophile is going to grow at a pH of somewhere around 1 to 5. These are microbes that you might find in the stomach, like H. pylori. They're microbes that you might find in the female lower GU, which can also be acidic. You find them in special environments where the water is acidic, usually around mining, runoff, and that kind of thing. A neutrophile, very similar to us because they're going to grow at a pH of somewhere around 5 to 8. Neurophiles are typically, going to be microbes that associate themselves with the human body and freshwater systems, that kind of thing. Our body, for the most part, again, except for the stomach and the female lower GU, is really buffered right around a pH of 7.2. Then you've got the halophile. These guys like a higher pH, somewhere around 8 to 12. Once you get too far beyond 12, they really struggle to grow, but these are going to be microbes that like those higher pHs. Next way that we classify microbes is based on salt concentration. You see that halo right there. Halo means salt, so halophile would be salt-loving. Now, there are non-halophiles. These are organisms that don't like high salt concentrations. We're a non-halophile, by the way, so we don't want high salt concentrations either. And so the microbes that are going to be found in these environments are typically going to require salt concentrations of around 1 to 1.5% or less. They get too high, you wind up with a hypertonic environment, something we talked about in a previous video. They can't handle that because they are losing too much water to their environment. They shrivel or they go through plasmolysis. Just as a point of reference, we human beings, we have a salt concentration in our body of roughly 0.9% sodium chloride. There are some exceptions on the skin, which we're going to talk about in a second, but internally, we're at about 0.9%. And so the microbes that would be growing in our body would also usually be non-halophiles. They could be halotolerant, so these are salt-tolerant organisms that can tolerate salt concentrations up to 12%. Again, that depends on the species. The halotolerant microbes are often going to be microbes that grow on the skin. A lot of our staph microbes, like Staph epidermidis, Staph aureus, that grow on the surface of our skin are halotolerant. They can withstand those high salt concentrations. They don't need them. So the salt-tolerant, halotolerant microbes can certainly grow in high salt concentrations, but they don't require it. That's different from the halophiles, salt-loving. Halophile, you're going to see that there's a lot of overlap in terms of their salt concentration range. The difference is our halophiles require those elevated salt concentrations. And so a really good example would be the microbes that you find in the ocean. The ocean is salty. It's got 3.5% sodium chloride solution or sodium chloride concentration. Those are going to be halophiles. The extreme halophiles are microbes that you would find in, well, extreme environments because they can handle salt concentrations as high as around 35%. Again, not all extreme halophiles can handle salt that high, but some of them can. And so a really good example of this, let me show you, is this. This is a salt lake, and there's not a whole lot growing here. For a long time, scientists thought that these were sterile environments. They're not. There are microbes, there are halophiles here, extreme halophiles that can grow in these very, very high salt concentrations. And if you actually lower the salt concentration, they die. The solution becomes too hypotonic for them. All right, so now that we've got the environmental classification covered, let's talk about one more way to classify microbes, and this is based on their energy and carbon sources. This is going to be hugely important because once we start talking about microbial metabolism, we're going to be using these words a lot. So this is kind of an introduction to these words. And so when it comes to energy source, we're looking at two main categories. We're looking at organisms, not just microbes, but organisms that can use visible light to extract energy from other substances. That would be a phototroph. Troph means to eat. Now, a phototroph is not using the light as the energy source. It's using the light to release energy from other substances, something, again, that we're going to talk about in a future video. The other option is that you have an organism or a microbe that is using chemicals as a source of energy. We call those chemotrophs. Now, if the organism is using chemicals as a source of energy, we have to define what that chemical is. And so it could either be a chemical source of energy, but that chemical is organic, so chemoorganotroph, in which case that microbe or that organism is using carbs, lipids, or proteins for a source of energy. That's us, we human beings. We are chemoorganotrophs. There are microbes that do the same thing. The other option is chemolitho. Litho means rock, and so these organisms are using not necessarily rocks, but they're using inorganic substances as an energy source. So not carbs, lipids, and proteins. Examples, and there are lots of them, but some examples would be things like ammonia, nitrite, sulfur, hydrogen. That's what they're eating. That's their energy source. And so again, we either have photo or chemo. And if it's a chemotroph, we have to define what that chemical is. Is it organic or inorganic, litho? And then you have the carbon source. And so we define what are the microbes using for carbon. Carbon is different from energy. Carbon source, the carbon is being used to build structure in these organisms, proteins and lipids and that kind of thing. And so cytoskeletal elements, using it for growth. And so while energy is used to perform metabolic tasks, microbes and things can move around and people and all that kind of thing, but the carbon is structural. And so for the carbon source, it's either going to be hetero or autotroph. A heterotroph consumes carbon. We, human beings, are heterotrophs. We have to consume our carbon from once living things, while autotrophs can fix CO2. In other words, they can take CO2 from the atmosphere, bring it in, and start to make organic compounds. Organic compounds are carbon-based, something we learned about in a previous video. CO2 is not a organic compound because organic compounds usually have hydrocarbons, carbons and hydrogens, and usually multiple. But CO2 is inorganic, and they can take that inorganic CO2, fix it, which means turn it into organic compounds, things like proteins and energy sources. So some examples here. We can actually combine these words together for both energy and carbon. And so you could have a photoautotroph. And your job, if you really want to understand this, is to learn how to break these words apart. Photo means that we have an organism that was using visible light to extract energy from some kind of a substance. Auto means it's fixing CO2. This would be a plant. There are microbes like cyanobacteria that are like this as well. Algae can do this. We have photoheterotrophs. This is weird, something we'll talk about in a future video, but this is an organism that's using the visible light as an energy source, but actually gets the primary carbon from something that was once alive. Then we have the chemoorganotroph, chemoorganoheterotroph. That's us. Chemical source of energy. that is organic, carbs, lipids, proteins, and we also have to consume our carbon from other once living things to grow our bodies. There are microbes just like this as well. And then we have the chemolithoautotrophs, chemical source of energy. That chemical's in organics, like the hydrogen or the ammonia that we were talking about before. And they're also autotrophic. They fix CO2. Really important microbes at the base of the ocean next to thermal vents. They are actually the primary producers down there. So now that we understand all these environmental or at least we've talked about the environmental classifications in the food, we can actually have a pretty good idea of how microbes grow in a closed system, in a lab, in a broth tube especially. And so this is showing us the growth curve. This is what microbes do. There's usually a lag phase. So when you put microbes in a liquid broth, it's going to take some time for them to grow and get used to their environment. Maybe they've got to turn on some enzymes or turn on some genes that produce some enzymes to help them exploit a food source. And so there's going to be a bit of time where they're getting used to the environment. And then once they do, they take off. They grow like crazy. That is the log or exponential phase of growth. And importantly here, if you want microbes to produce something for you, maybe it is some type of a chemical. Some microbes can produce, they're genetically modified and they can produce insulin or growth factors. I was recently at a place where they grow microbes and sell the microbes so that they can basically consume some of the products of fish waste, like these fish tanks behind me. And they are constantly trying to keep the microbes in this exponential phase to produce as many microbes as possible so that they can bottle them up in a buffer, sell them to people who have fish tanks so that they can control some of the issues that can happen in aquariums. And so this log phase is really important. But then what will happen is the microbes grow like crazy. They've got unlimited space, they have unlimited food, there's not a lot of waste, and so they're just growing, growing, growing until there is a limiting factor, and that's our stationary phase. The number of microbes growing is now equal to the number of microbes dying. They're starting to struggle a little bit. Maybe there's more food competition for space. Waste products are starting to accumulate, and so the environment is less than ideal. And then, if left alone to continue, they will go through a death phase or a decline phase where there's just too much waste, there's too many microbes, there's not enough food, and so there's a real struggle for existence, and that death phase can happen for days, weeks, even months before all the microbes perish. Because eventually what happens in the stationary and death phase is the microbes start consuming the organic compounds from the other microbes that have lysed and died. Now it's important to note that that's what's happening in a closed system in a lab. In nature, this is not necessarily the case because we have an equilibrium more times than not. So in nature, microbes actually grow much slower because they're not giving the ideal environmental parameters like we do in lab. If we know a microbe likes a certain temperature, likes a certain food, we give that to them. But in nature, there can be a struggle for existence where the environmental parameters aren't ideal. Maybe the temperature is not quite right. There's not enough food. And so maybe they don't grow and divide as fast. And so they reach kind of a stationary phase and they're in a much larger community. And so in a lab, usually we're dealing with monocultures. And so if we're growing an E. coli culture, it just grows. If we're growing S. marcescens, that's the only thing in the tube. But in nature, microbes are competing with one another in a population, but also amongst all the different microbes that are in that environment. And so generally speaking, there are two main types of media that we use in microbiology. One is called a broth, and we can see the broth here on the left-hand side. That pretty much looks like a mostly sterile broth. On the right-hand side, you can see it's very, very turbid. It's very cloudy, and that's usually what it looks like after microbes have been growing for a long period of time. When it comes to liquid media, usually microbes are going to grow really fast in liquid media, but they're also going to die back very quickly because they're surrounded by the nutrients. So they grow fast. Generally speaking, they die fast as well. The other type of media is called agar. And with nutrient agar, what we have this. That's a nutrient agar plate. We also have the tubes, which I showed you previously. Nice thing here is we've got all the nutrients within the agar. The microbes generally grow on the surface. And we can see here, we've got all these things called colonies. Those are the circles that you see. And there's different types of microbes here. You've got some yellow ones. You've got some that are kind of off-white, some that almost appear clear. And so what's nice about the solid media is you can see colony characteristics. You can see color. You can see texture. You can see the size. And so this is really, really cool. Now, one thing here is that you are limited by space. As the colony grows out, as it touches another colony, it's going to have to stop. And so with the colony characteristics, What's interesting here is that you've got the ability to see different types of microbes, and each one of these colonies was derived from a single cell. Now, the nice thing is once we know how microbes grow, we can actually use something called differential testing to determine what properties microbes have and even identify microbes when we put enough of these differential tests together. Differential tests allows us to tell the difference between different species of microbes. And so this is a really good example. There is a dye here. This is most likely phenol red. And what's happened here is the phenol red changes colors depending on how the microbes use the media. Phenol red is a pH indicator. And so in this particular example, if we have orange, generally speaking, that is going to be slightly acidic. Pink is going to be basic, and yellow is going to be very acidic. This stuff actually usually starts out red. And so it's a differential test because the media reacts to what the microbes are doing. The microbes impact the media. And there's also something called a selective test. In selective media, what's happening is the media impacts the microbes. It's the opposite. And so the media is either going to allow the microbes to grow or not grow. This is a citrate test. And while there is a color change here, the color change is really just helping us see whether or not the microbes are actually growing. And so with the citrate test, the whole point is, can the microbes use citrate as a carbon source? If they can, it turns the media blue, but you really don't need the color change. You just need them to grow. If they can't, the media stays the same color. Another interesting fact about media is it can either be complex or chemically defined. And what that means is complex media is pretty much a lot of what we use in microbiology. It's got a certain percentage of peptides or yeast extract or beef extract. In other words, we know that there's stuff in the media that microbes generally like to eat and they grow fairly well, but we don't necessarily have every single percentage of every single component in that media nailed down. Chemically defined media is a little bit different. With chemically defined media, we do know the percentages of every single thing that's in that media, and it can be really, really useful if you're running experiments that require you to nail down the nutrients so that it is not fluctuating, or one of the nutrients is something that you're going to be experimenting with. You can use chemically defined media to alter just that one aspect. Where complex media, it's less expensive, it's a little bit easier to produce, but you can't really run the same level of experimentation as you can with chemically defined media. While we're talking about microbial growth, it is important to note that some microbes can form colonies that can produce biofilms, and biofilms can be a very important aspect in the biomedical world and medicine because this biofilm, its whole purpose is to protect bacteria. And so you can kind of see the process here where you've got microbes that attach to a surface and then they begin to secrete the biofilm. I want you to think of the biofilm as kind of like a protective shield, and that protective shield protects the bacteria from other microbes, predators, our immune system, chemicals like antiseptics, disinfectants, antibiotics. And so when biofilms start to form, it can create all kinds of problems for us treating bacterial infections. And here's a really good example of a biofilm. This is the tartar that forms on the teeth. And if you let it go this long, yes, microbes will continue to form those biofilms and they will get larger and larger. That's why people go to the dentist. They get that biofilm scraped off. They get their teeth cleaned. Because if you don't, you wind up with some serious gum issues. Last thing we're going to talk about in this video is, okay, if we know that we can grow microbes and how to grow them, how do we quantify them? We talked about that formula, but that's not really showing us in a lab how to quantify bacteria. How do we know how many cells are present? And there's lots of different ways to do this. Before I get started in the different types, understand there's usually three main concepts that I want my students to understand. And concept number one is, when choosing a quantification method, how much time do we have? Do we have a day? Do we have a week? Do we have just hours? How much money do we have? In other words, do we have money to use expensive equipment, or are we going to have to do this in a very less expensive way? And then, so we've got how much time, how much money, and then the third thing is, do we need viable cell counts? Because only viable cells cause disease, or can we do total cell count, which includes the cells that are dead? Okay, now that we know those three things. All right, so this first one is called a slide count method. The slide count method works because it has a fixed volume, so you know your volume, and then it also has these gradations here that tells you what the volumes are for each size of square. Now there's some downsides here. The downside is you cannot distinguish between living and dead cells. And so unless you've got some superhuman ability that nobody else has, and if the cells were for some reason motile, that's out. It is cheap and it is quick. And so those are the upsides. But the downside is, again, it's really going to be difficult to distinguish between living and dead cells. And in terms of reliability, again, we're dealing with incredibly small volumes on these slides. And so you have to do a number of different counts. And sometimes you can get very significant results depending on your sample. Another quantification method is turbidity. Now, with this particular method, it's more qualitative than it is quantitative. And what I mean by that is you're looking at turbidity. You're looking at the cloudiness of a tube. And obviously, the more cloudy a tube is, the cloudier a tube is, the more microbes grew. And so I'll give you an example of how we use this in our biology labs. Sometimes we will grow microbes to see if they like certain sugar concentrations or if they like certain temperatures when they are going through fermentation. The cloudier the tube is, the more they like that environment. Now, can we tell exactly how many microbes are there? Not really. However, we can say, okay, well, this microbe preferred 10% glucose over 10% sucrose because the tube is cloudier. And so it's a very simple way to see how microbes prefer a particular environment. And you can actually get pretty technical with measuring turbidity. It doesn't just have to be an eye test. You can use something called a spectrophotometer. And the spectrophotometer will actually put a measurement on the amount of absorption a solution has. You can also use membrane filtration as a quantitative method. Now, this is interesting because you can run a volume of fluid, water, solution, sample through a filter. That filter is going to catch microbes. And so you know how much in terms of volume, how much you ran through that filter. Now you have to take that filter paper and put it on a nutrient agar plate. And then every one of the colonies that you see here was derived from a single cell. And so then you can get the number of cells based on the quantity that you pass through the filter. Nice thing here is you're only getting viable cells. So because you're only getting viable cells, that's nice. It is very cheap to run, but unfortunately you're going to need at least a day to incubate this. And by the way, just like with turbidity, you're going to need at least a day to incubate that as well. But it is cheap and it is fairly simple. Another way to quantify microbes, and this is one we do in our labs, is called solution dilution. And so what we do is we take a sample, a tube that's got microbes in it, and we dilute it. And we do it serially. That's hard to say, serially, but we serial dilute it. And so we know our concentration, our dilution concentration, and then we plate that. We take some microbes from that solution and put it on a nutrient agar plate, and then we can count the number of cells on that plate after a day or two, and then we'll know this number of cells equal this dilution factor. We do some math that I talk about in one of our labs, and then we can pretty much get an answer as to how many cells per milliliter were in that solution. So we're getting viable cells. It is certainly inexpensive. It does take a day or two, so that's a downside. And again, the results here, when my students do it, and they've all done it just one time, but sometimes we'll see that even though all my students take the exact same tube, take samples from the exact same tube, we get some different results, sometimes orders of magnitude different. One other thing you can do is you can measure metabolic byproducts. And so you can measure things like pH. You can measure gas production. That's this little Durham tube inside these tubes here. These smaller tubes are trapping gas. And again, for this part, it's more of a qualitative thing. It's more of a comparison than it would be just quantifying like, oh, I've got 6 billion cells in here. But you can compare more gas or more acid or more turbidity. You're getting more growth. The microbes are happier in that environment compared to the one that has less. One of my favorite methods is this one here. It's called the flow cytometer. Now, unlike everything we've talked about so far, this is expensive. This is going to be by far the most expensive thing you can do. However, you can get viable cells from this, and it can be one of the most efficient ways to do so. And the reason for that is this flow cytometer, very sensitive machine. You have microbes that can be fluorescently labeled if they're alive, and they pass through this flow cytometer single file. And then there is a laser that hits these cells, and if they have absorbed that chemical, that's going to allow the detector to detect that as they go through. The laser is going to hit it. It's going to fluoresce. On the other side, there's going to be a detector. It's going to go boom, boom, boom. It's just going to count those microbes as it passes through. So very expensive. You can get viable cell counts. You can get them faster than just about any method that I've talked about so far today. Downside is, again, it's the expense and the maintenance. And so the maintenance here, because again, you're dealing with tubes that are allowing single microbes to pass through. If you forget to switch them out, because sometimes you want to do eukaryotic cells, sometimes it's prokaryotic. And so you really have to know what's in that solution and what's going to be passing down that tube. Because if it's too big, the cells are too big, it's not going to pass through. And if they're too small, you might wind up getting multiple cells passing through at the same time. All right, everyone, I know this was a little bit of a longer video, but now you have a basic understanding of how we classify microbes based on their growth, how we quantify them. As always, thank you so much for being here. Hope you have a great week.